So thanks everyone for joining the Pheo Para Genetics Breakout and for sending questions ahead. I prepared some slides based on the questions that were already sent in, and then feel free to put any questions in the chat as well. Okay, so there were a couple questions surrounding genetics of Pheo Para and genetics in general. So I wanted to take a minute to talk about genetics. So when we talk about genetics, we're really talking about your DNA. And DNA is the genetic code determining characteristics of a living thing. And it looks like a helix, a double helix. This top picture here, I really love. Maybe not exactly what it looks like in our cell, but I like that picture. DNA kind of looks like a twisted ladder. And the rungs of these ladder are made up from 4 types of blocks, which we call bases and base pairs. And there are 4 base pairs, A, T, C, and G. And there's about 3 billion of those that write our human genome. And the sequence of those blocks of those letters make up the instructions of how our body functions. And then we take that sequence and we break it up into 3-letter words, or we call them codons. And each of these 3-letter words then get strung together to make a sentence or a gene. Now, a gene is the part of the DNA that tells the cell how to make a specific protein. And then that DNA gets translated into a message, which is called RNA. And then that message gets translated again into proteins. And there are tens of thousands of proteins that build our human body. And proteins are the workhorses or the machinery of our body that helps us function. So all of us are unique. We all have very similar DNA. That's what makes us human beings, but there's slight variations, which makes each of us individual. So let's talk about the variations in DNA. Those slight variations, the way I like to think about them and that I explain to my patients in the clinic, is that differences in our DNA can be thought about in the differences between the American versus the British spelling of a word. So theater versus theater with an RE, or labor versus labor with an OU or just the O. They mean the same thing despite the slight variation in how it's spelled. And we can also have variations that change the meaning of our words. So the example there is to take the word state and make it slate. So 1 letter change, but has a very big meaning difference of the word. Or eat versus seat. Again, 1 letter inserted, very different meaning. So that variation, when it doesn't change the meaning of the word, we call that a benign variant. And we all have lots of different benign variants between us. But a pathogenic variant or a mutation is when that change in DNA really changes the meaning of the word. And so when we talk about genetic mutations or variants which lead to disease, we're talking about the pathogenic variants or mutations. Now, there's one more difference that's important to keep in mind, and that is the inherited DNA variants versus the tumor DNA variants. So when we talk about inherited variants, we're talking about the germline variants. Those are the variants, the changes in DNA that we're born with. A lot of them are benign, and occasionally there might be one that's a pathogenic or a mutation. These variants are found in every cell of our body because we're born with it. We start as 1 cell and we multiply and multiply until we get into our human form. These are usually tested through blood, sometimes through saliva testing, and these are the inherited variants that can lead to predisposition for developing tumors. On the other hand, we can talk about tumor-specific variants. These are called somatic variants. This is when we're already a full human, but there's a DNA change in 1 cell of our body, and that change proliferates and proliferates. The cell grows and forms a tumor. So there can be a variant in the DNA that's just in the tumor cells that's not in the rest of the body. And sometimes we can take advantage of that to use it for treatment purposes. Okay, so that was the section to understand genetics. Let's take a brief look at pheochromocytoma and paraganglioma. Probably everyone watching this knows what they are, but just in case, Pheochromocytomas are the neuroendocrine tumors within the adrenal glands. The adrenal glands are those little hats that are sitting on top of the kidneys. And then paraganglioma can be anywhere from the base of our skull all the way to the pelvis, and they form in the little nerve bundles or ganglia throughout our body. Pheos and paras are unique in that many of them make hormones, and they make adrenaline hormone. And the fancy medical term for adrenaline hormone is catecholamines and metanephrines. These are our fight or flight hormones. So if we look at the adrenal gland, which you can see up here, or the ganglia, the nerve bundles, which you can see on the left side of the screen, adrenaline hormone can be made from either of these. They bind to a receptor and lead to our fight or flight response. That's the response that we need if someone scares us. Our heart starts racing, we become sweaty, our eyes get really wide, we're prepared to run away. But if you have a tumor in the adrenal gland, a pheo, or a tumor in the ganglia, a para, this can lead to constant adrenaline production, which can cause high blood pressure, rapid heart rate, sweating, headaches, anxiety, tremors, and increased blood sugars, among other symptoms. Now, some people can have high adrenaline and not recognize it, so they don't feel any symptoms. And then there are some paraganglioma that don't make adrenaline, and so they don't lead to these kind of symptoms either. The way that we test for a secreting Pheo Para, one of the tumors that does make these hormones, is to test for plasma metanephrines. And sometimes people will test for a 24-hour urine collection for metanephrines. Either one of those two tests is fine to do. In my practice, I start with the plasma because that's a blood test. It's a lot easier than collecting urine for 24 hours, but it can be done to collect urine as well. Okay, one of the questions had to do with what is the chance that my children might get a genetic component of these tumors. So it turns out that Pheo and Para are highly hereditary. So up to 40% of all patients with Pheo Para have a hereditary component, meaning they're born with something, some change in a gene that predispose them to get a Pheo or Para. And if a child has a Pheo or a Para, then up to 80% of those can be hereditary. Turns out it's not just one gene. There are many, many, many genes that contribute to Pheo Para. In fact, from the early 90s, there were 3 genes that were recognized, NF1, RET, and VHL. But in the last 2 decades, many more genes have been found. And here on the right are a list of some other genes that are suggested, perhaps not yet fully proven, but highly suggested to be associated with Pheo Para. So there are many genes that contribute to risk of developing Pheo or Para. And each of these genes is associated with a different syndrome. So this was one of the other questions submitted today, which is, what are the syndromes associated? So let's start talking about von Hippel-Lindau disease. Von Hippel-Lindau affects about 1 in 3,600 individuals. And it's a tumor syndrome that the patients are at risk for developing lots of different tumors. Doesn't mean that every patient will get all of these, but they could get kidney cancer or cysts on the kidney, hemangioblastomas, endolymphatic sac tumors, and pancreatic neuroendocrine tumors. They're also at risk for pheochromocytoma. About 20% of patients with VHL will develop a pheochromocytoma, the ones usually in the adrenal gland. The next syndrome is neurofibromatosis type 1. This is caused by changes, pathogenic variants in the NF1 gene, affects about 1 in 2,800 to 3,500 people. Patients can have lots of different things associated with NF1, cafe au lait spots, neurofibromas, which are peripheral nerve sheath tumors that can be anywhere in the body, and lots of other things as well. Pheochromocytomas are not part of the clinical criteria for diagnosing NF1, but they can occur at an increased frequency compared to the general population. About 1 to 13% of people with NF1 will develop a pheochromocytoma. There's also some risk for pancreatic neuroendocrine tumors in people who have NF1 as well. The next syndrome to talk about is multiple endocrine neoplasia type 2, caused by pathogenic variants in the RET gene. It affects about 1 in 40,000 individuals. And there are 2 types, MEN2A and MEN2B. In both types, the risk for medullary thyroid cancer, which is another type of neuroendocrine tumor, is almost 100%. In MEN2A, which is more common, about 30% will develop hyperparathyroidism, which is a condition that regulates calcium incorrectly. And about half the people with MEN2A will develop pheochromocytomas. MEN2B is similar, but patients are not as at risk for the hyperparathyroidism part of it. But again, overall, about 50% of people with MEN2 will develop pheochromocytoma, mostly in the adrenal glands. The next group of genes is associated with a syndrome called hereditary paraganglioma pheochromocytoma syndrome. And there's 5 different genes associated. SDHB, C, D are the more common, but also SDHA and AF2. The risk for developing a Pheo or Para with any of these gene mutations is different, which is interesting because all these genes are cogs in the same protein complex, but the risk of developing Pheo Para varies depending on which gene is affected. With SDHB, the risk of developing a Pheo Para is about 25% by age 60. And that's compared to, say, SDHD, where the risk of developing Pheo Para is about 45% by age 60. And both of those contrast SDHA, where the risk is quite low of developing a Pheo Para. It's 10% by age 70. So for the person who asked the question of what's the risk for my child, it really depends on which gene that person may have. If that person does not carry a variant in any of these genes, the risk to the children is the same as the general population. But if that person with a Pheo Para has a genetic variant in one of these genes, or the prior ones I talked about, the risk to spread that to their children, or I shouldn't say it that way, for their children to develop a Pheo Para is about 50%. But if that child has a 50% chance of getting that gene, their risk of developing a Pheo Para is different depending on the gene. So it's a little bit of a complicated answer to the question, and it really is gene specific. So I encourage everyone to speak with their doctors about that based on their own genetic risk. Now, I'll mention one other thing about SDHD, D for dad, and that is because SDHD and AF2 are paternally inherited. That means if you inherited this gene variant from dad, you are at risk to develop Pheo Para. But if you inherited the gene variant from mom, you carry the variant, but you most likely will not get a Pheo Para. You still have a chance of it being passed on to children through the DNA, but you yourself wouldn't develop a Pheo Para if it came from mom's side, with very rare exception. The last thing I'll mention about the SDH genes is that they also increase risk a little bit for kidney cancer and GI stromal tumors, and perhaps pituitary tumors and others. Now, there are several other genes which have been identified. I'm just listing some here. They all have associated syndromes with them, but Pheo Para is the most common finding with them. But there can be other tumors and things associated, and I'm happy to take any questions about that. So should you be tested if you have a Pheo Para for any of these syndromes that I talked about? And the answer is, in my opinion, yes. The guidelines from multiple different societies, the Endocrine Society and the North American Neuroendocrine Tumor Society and others, recommend that all people who have a Pheo Para be referred for clinical genetic testing. And the reason is, as I mentioned, up to 30 to 40% have a hereditary cause for their tumor. So we do recommend testing everyone. And the question is then why? And the answer is because it helps us guide how we should screen that person and monitor them going forward. And it also helps determine what their family member's risk is. So if that person has a Pheo Para but never doesn't have a genetic cause, we monitor them in a different way than if that person has a Pheo Para and has a genetic cause. There may be other things we need to do for screening. So it changes how we manage you and your family members. Now, again, keep in mind that every person that has an inherited gene mutation to develop a Pheo Para are not going to necessarily get the features. So just because you have a pathogenic variant in one of these genes doesn't mean that you will develop a Pheo Para or one of these other tumors. But we have screening guidelines for what to do if someone does have that gene variant and hasn't had a tumor yet. There are certain recommendations that are different for each gene of how often we should screen them and how we should screen them. It usually involves blood tests and imaging. Let's just spend a minute talking about metastatic disease because I got some questions about that. It can occur with Pheo Para. It's not common, but it absolutely can occur somewhere between 10 and 25% of the time. When there is metastatic disease, what we mean is that the cancer now has spread to other parts of the body. And the most common areas are bone, lymph nodes, lung, or liver. We don't have treatments that can cure metastatic disease usually, but we do have a lot of treatments that can control the disease and get it under control for the patients. And any person who has a Pheo Para should have lifelong follow-up so that we can monitor for any sign of a recurrence or metastatic disease forming. There's lots of research that's really needed to help us really treat and understand who's at risk for metastatic disease. We do that through registries, surveys, patients offering to give blood or tumor samples. There's a lot of clinical trials going on for diagnosis and prediction, as well as therapies. People are trying to develop animal models and cell lines. So it's important to participate in research when and where you can if you're comfortable with it. The reason is that many of us have this goal of precision medicine, to take Pheos and Paras and really try to treat the individual at the time for their particular risk of disease. So we used to treat everyone the same way, but the idea behind precision medicine is to try and understand whose tumors are going to respond to particular treatments and treat people as individuals based on that. So it's moving from a one-size-fits-all system to a system of predictive, preventative, and precision care. So in the Pheo Para realm, there are a couple ways we can do that. We can look at that tumor DNA, like I talked about now, not the inherited genetics, but the tumor-specific genetics. And depending on the mutations in the tumors, we may be able to offer different treatments to be more effective for that particular tumor. There are other examples in Pheo Para of how we do precision medicine. One of those is with the radionuclide therapy, which you heard about earlier in this conference. The tumors themselves must be avid for treatment. So if the tumor picks up MIBG or picks up dotatate, then we can offer them MIBG therapy with a Zedra or dotatate therapy, which is Lutathera or PRRT. We're doing research now to understand genetic markers of who might best, respond within those groups in particular. And can we find tumor markers that will predict who will develop metastatic disease? Okay, so we can also use the person's germline DNA to try and predict who's going to get disease and how we can prevent it. These are ongoing studies that we're doing right now. I have NIH funding to do this across 9 centers in the US and Australia to try and see if we can understand who's at risk. So again, I just want to say thank you to anyone who does participate in research for helping us address these questions. The last thing I want to say about the one of the other questions was what other treatments can be used if your tumors are not radioligand avid. And there are many clinical trials ongoing now to try and address that. A lot of the inherited mutations affect hypoxia-inducible factor, or HIF. And there's a new treatment called belzutifan, which is approved for certain tumors through VHL. It's now being studied for Pheo Para as well because of that activation of hypoxia-inducible factor. So there's a clinical trial, and hopefully this will be an approved therapy in the future. This slide is very complicated. It's my last one, but it's just to show you all the pathway information we know about Pheo Para so far, and we can take advantage of this for different clinical trials and therapies. Down here, circled in red, is where the HIF inhibitors are that I just talked about in the prior slide. And these are showing other areas where we can maybe use different types of drug therapies to treat metastatic disease in Pheo Para. And many of these are in clinical trials now. So thank you for your time. I hope I answered most of the questions sent in ahead. Thank you very much for your time and attention.